I'm Ben Raphael. I'm a professor in the computer science department at Princeton. It's great to be back at CGSI. I am very disappointed in the organizers in scheduling me right after Pavel, who's a very tough act to follow. Great communicator in live talks, as we've heard, a great communicator in YouTube videos, which many of you have seen, and in textbooks, which we also heard about. It's especially intimidating for me because I did my postdoc with Pavel, who brought me into computational biology and really taught me how to be successful in this field, how to write a good paper, how to give a good talk. And so if this talk is bad, we know who to blame. Now, a thing I learned recently, though, not from Pavel, in the past year or so, is that if you want to increase the interest in your talk, and I apologize that this was only posted yesterday, this title, so if you're expecting something else, I'm sorry. But I learned that if you want to increase the interest in your talk by about an order of magnitude, you just add on "with AI" on the end. And so I've done that, and there will be some AI, as you'll see. And so I should acknowledge really up front, you're going to see that the work I'm going to present is the work of Peter Halmos, a very talented grad student working with data scientist Julian Gold, and another one of my talented PhD students, Chen. All right, so the setting for this talk is really looking at one of, I think, the most fascinating and interesting questions in all of science, which is how does a single cell become a multicellular organism? And what are all the changes? And I see if I can click on this little video. Maybe not. Maybe I have to turn this off. You can see this little video as I talk. Yeah, there we go. So this is zebrafish development, very intricate series of cell division, obviously. Cells differentiate, cells migrating to create this complex organism. So I wanted to tell you about some of our work in this topic. And I did this a little bit last year, and I had this warning last year that I'm not a developmental biologist, so you should take everything I say with appropriate grain of salt. But I kid you not, I was working on these slides on the plane flight over, and this guy was sitting diagonally behind me. And when we got off the flight and we're all waiting to get our bags and stuff, he says to me, "Are you a developmental biologist?" And I was so jarred by this question. It's a very weird question for someone to ask you as an introduction. I was like, no, why would you ask me such a question? He said, "I saw you working on your talk slides. I couldn't help but look." And it turns out this guy's a retired medical oncologist, so a card-carrying MD thought I was a developmental biologist. So I think I've really mastered the art of what the young people call fake it till you make it. So here we are. All right, so my simplistic view of development, it's a superposition of three processes. Cells divide, obviously. They differentiate. Developmental biologists have a way of describing this, which goes back to Waddington, where undifferentiated cell sort of moves through this landscape of cell state, sort of as a marble rolling down this landscape and into these terminal cell types. And then cells migrate, form these very intricate structures. Now, as computational people, we can think of what are the things we need to represent these three processes. So division, cell lineage tree, very easy. Differentiation, some sort of map between these cell types called differentiation map. And then migration, you just need to know the XYZ coordinate of every cell at every time. So this leads us to sort of four key questions that each have a concrete computational question. What's the history of cell divisions? So that's reconstruct the tree. The cell type, cell states, at each time point. So you just want to group cells according to whatever measurement you have. What's the history of these cell divisions? So that's reconstructing this differentiation map. And then how do cells migrate? And you want to connect cells in space over time. Now, last year, I talked about ways to sort of answer maybe questions 1 and 3 using technologies that look at cell division by heritable markers, either natural markers or induced mutations. There's a field called lineage tracing. And so I haven't had a couple talks on this at CGSI, but it's a growing field. And there's been multiple talks at CGSI. I probably missed a few, so I apologize for that. And then I also talked about this problem of finding differentiation maps from this lineage tracing data. It's a method called CARTA, which I presented here before we'd submitted it, and that submission was very successful, and it's going to appear in Nature Methods soon. That's work of Palash Sashitel, who's sitting right next to Pavlov here in front. What I want to talk about today is more about this problem of migration and differentiation in space. And the enabling technology here is spatial sequencing, or often spatial transcriptomics that's being measured. And there are many such technologies, but roughly speaking, what they do is they'll take a tissue slice, a 2D slice, and then measure, in this case, RNA expression at thousands or tens of thousands of locations in that tissue. And so the data you get out is at every spatial location, in this case, the expression of every single gene in the genome. But corresponding to that measurement, you have the actual 2D location where you made the measurement. Okay, so this spatially resolved gene expression. A lot of development in the field, lots of technologies that have different spatial resolution, different number of spots, different coverage of how deeply you can measure the transcriptome. We're going to sort of gloss over those details for the most part in this talk. So what got us interested a couple of years ago was that we started seeing data sets where researchers were applying spatial transcriptomics at multiple time points during a developmental process. This is one from mouse embryonic development. These are the sort of days of development. And we thought, if we could identify at each time point the ancestor cell at the previous time point, this would give us information about all three of these processes: cell division, lineage, differentiation, because we could compare the cell type at the later time point to the previous time point, and then obviously migration, because we would see where the ancestor cell was located. Okay, so I should say that this is a destructive technology, so these are different animals usually that you get these measurements from, but we can assume that development is very stereotypical, so we could find these correspondences across different animals measured at different time points. So how exactly might we address this challenge? And so we could think sort of simplistically that development from this data is just described by a map. And this map takes this measurement of cell state, gene expression, or maybe it's chromatin or whatever we have measured at these XY coordinates at one time, and then it moved them forward in time to the next time point. Okay, so if we could infer this map, well, if we reversed it, then we find the ancestor, and I reversed it because each cell should have a unique ancestor, but they don't have unique descendants. Differentiation, we could look at how far in cell state have we moved during that time, and we can, of course, look at migration. How far have we moved in physical space? So this leads to the problem of how can we infer this map from this spatial temporal transcriptomics data. And when you think about this, the first thing you might think is that, well, okay, one, it's hard. Two, it's a completely underdetermined problem. Just giving you these two time slices. And so I'm asking a lot from these two time slices. So it's an underdetermined problem, and an approach that's been used by us and others is a technology and a method called optimal transport. So what I wanted to do today was first tell you a little bit about optimal transport, and then describe a few of the ways that we've extended optimal transport and used it in different ways to attack this problem. All right, so overview of optimal transport. Let's begin with just a simple puzzle, not due to me, due to Marco Couturi. He's one of the real leaders in this field. So I've got a set of data points that I've measured, and I measure them. There's this light. I turn off the light, and then I turn it back on, and the points have moved. Okay? So the question is, where did each point go? What's the correspondence between the two sets of points? Now, there's really no answer to this question. Any point could have gone anywhere. It's completely underdetermined. So in order to make this a realistic problem that we have a hope of solving, we need to impose some cost. Okay? And so maybe a natural thing to do would be to say, well, maybe each point hasn't moved that far. So let's find the map that minimizes the amount of movement in space. More generally, we could think of defining a cost between every point and every other point, and we could try to find the minimum cost explanation for the movements of these points. And that's exactly what optimal transport is, just finding these minimum cost maps. And there are two different approaches that have been taken to this problem. The old version, and I didn't put all the references here, I really didn't learn well enough from Pavel all the great history. I should have put the pictures of the individuals that worked on this, because this Monge problem goes back to the 18th century from Monge, who was interested in finding these bijections, so one-to-one onto correspondence between these sets of points of minimum cost. And this is a form of a matching problem. You can actually solve this with sort of classic matching techniques. If you do that, it turns out to be cubic, and in space quadratic. This will come back later in the talk. And this cubic scaling doesn't work so well when our data sets have thousands or tens of thousands of points. So the big innovation here was to sort of relax this problem and allow for partial assignment of points to each other. Okay? So via this coupling matrix. So this is a doubly stochastic matrix. Each point can be proportionally assigned to every other point and vice versa. And so this relaxation, if I just write it down this way, we didn't really gain anything. It's still cubic time to compute. But the real innovation here in 2013, Marco Couturi realized that an earlier algorithm, due to Sinkhorn, could be adapted to solve a slight variant of this problem. And in the variant of the problem, you add this additional term to the cost related to the entropy of that map. So you can just compute the entropy of this coupling matrix. And this entropy regularized optimal transport with the Sinkhorn algorithm can now be computed in quadratic time. And this is a big gain from cubic to quadratic time, really enabling this to be applied to thousands or tens of thousands of data points in a practical way. Now, some of you that have seen this before, or if not, I'll sort of show you. There's another way you can kind of think about this. If each point in space, you can actually just convert into some discrete probability distribution. I could assign weights to each point or even equal weight to each point. And so the other way to think about optimal transport, the sort of equivalent way, is that it's a mapping between two probability distributions. Okay? And if you think of a probability distribution as like a mound of sand, which you might see tonight in Santa Monica, then this is a minimum cost map from moving one mound of sand to another mound of sand. So this one version of optimal transport is equivalent to what's called the Earth mover's distance, the minimum amount of work you need to transform one into the other. All right, well, I mean, this sounds, I don't know, kind of boring in a way, mathematical, overly mathematical. But this idea of transforming probability distributions into probability distributions is an incredibly powerful idea. In fact, it's such a powerful idea, it underlies a lot of what is happening in generative AI these days. Now, people have different ways of computing these maps, diffusion, flow matching, but some of these are actually enabled or improved by using these techniques from optimal transport. All right? So, when you're generating a face from another face, it's really just computing maps between probability distributions over images. And this generality of probability distributions to probability distributions can also be used for many types of genomic data. And so optimal transport has found many uses in genomics. Maybe one of the first was if you think about single-cell RNA-seq data sets from 2 time points, you can describe the distribution over cells at those time points, map between them. It's a method called Waddington-OT, single-cell to spatial, NovaSpark. Ron Zier in my group did one about aligning 2 spatial data sets. Rita, who's here, did one on multimodal optimal transport, and many, many others. All right, so I'm sure I'm forgetting a bunch. So now I want to dig in a bit to 2 of these that are related to this problem of development. All right, so first is Waddington-OT. So we've heard Waddington already, the Waddington landscape, and this method was named because it was really their way of describing this mathematical model of the landscape. So thinking again about differentiation, this multipotent cell up here rolls down the landscape and can differentiate into different cell types. We could describe that process by thinking about the distribution over cell states that we observe at different times. Now, there's a figure from their paper, and they've reversed the landscape going bottom up here. But maybe at the first time point, there's just sort of this nice unimodal distribution. There's a single cell state with some variation, either biological or measurement. And then over time, if we measure at a later time point, we might see 2 states starting to emerge, which then lead to this bimodal distribution. So we measure these distributions at different times. Single-cell RNA-seq gives us this distribution over cell states, and then we just want to find the correspondences between them. All right? And this is what Waddington-OT does, and we can now write it all down if we want. And there's a slight shift in notation here because they used p for the data. So here it's Greek p or pi for the map. Sorry about that. But again, it's the minimum cost map, and there's these constraints that say every cell moved somewhere. Right? that's sort of what these constraints are. And then the other thing, and this is kind of the part I wanted to mention quickly, the cost matrix. So what's the cost of moving a cell from one location in gene expression space to another? Well, we can represent that through, say, Euclidean distance in some latent cell space. So if we do your favorite dimensionality reduction and then compute distances in that gene expression manifold. Okay, right, very straightforward, very simple. This is 2019 technology. One thing to immediately think about, if you think about development, you think about that movie I showed you, is that development also occurs in physical space. So it's not just about this marble moving through gene expression space. Cells are occupying some location in physical space. So how does this play out? If we just think about this cell state distribution and then moving this pile of sand to this pile of sand, what happens when we think about location? So let's do a simple version where we have a one-dimensional organism. Okay, so I could represent the location of each cell that I've measured, and maybe the cells at time point 1 are spread over these locations, and then at time 2 they move to these 2 locations. If I compute the minimum cost map just looking only at the expression coordinate, then maybe I get some correspondence like this. And then if we start looking at how cells have moved between the times, we see that there's a lot of movement. Every cell's moved everywhere else because we haven't tried to track location. And maybe if we had tracked location, we would find actually that we could not have the cells move at all, but just move just a little bit more in expression space. Okay? So this shows that keeping track of where the cells are is extremely important if we want to think about modeling development. So several years ago, we sort of extended this idea of optimal transport to spatial transcriptomics. And the idea is quite straightforward. Now, when you know where cells are, you look at your cost map. Here, pi, again, is the map. The cost, there's that gene expression cost, how far you move in gene expression space. But then now there's a spatial cost. How far did you move in physical space? And to make this computable, we used an extension called Gromov-Wasserstein optimal transport. Details don't matter so much. It's just a way of representing these distances where we described how far you move, not for one point, but by looking at pairs of points. So we try to minimize the difference in distance between mapped pairs of points. Okay? So we did this across 3D space. We did this across time. I described the generalization for time in my talk at CGSI last year from this same podium. All right, so now what? Well, now data sets have gotten a lot bigger, a lot better, a lot higher resolution. So if we look at a modern data set, like this mouse embryo data set that I showed you, here is spatial transcriptomics from mouse embryo. It's colored by the different cell types that they annotated. There's 77,000 spatial locations in this data set. At the next time point, there are 102,000 spatial locations. So if we want to compute this map from cells to cells, it's a huge matrix, 7.8 billion entries. So even a quadratic algorithm is not going to be able to compute this map at full cell resolution. All right, so what can we do? Leads to our next part, sort of already the hint. We look at low-rank optimal transport. So what is this about? So one way to scale this factorization is to actually not try to compute the full matrix P, but to represent it in a lower rank representation. And if you've done any work here or listened to enough talks here, probably you've learned about non-negative matrix factorization. So P is a non-negative matrix, and so we can break it apart into these 2 factors. The cells by sort of factor or signatures coordinate, which is much smaller, and then the matrix H, which describes the combinations of those coordinates. And so we can apply matrix factorization, non-negative in this case, since the entries are positive. So they're all these weights. If you haven't learned about non-negative matrix factorization, these days it's very easy to go learn something, right? You just can search it. So if we type it into Google, they have this great AI overview, and it tells us about what non-negative matrix factorization is. It's great. It even has this handy picture. And if you look in this picture, you get this nice illustration of it, except it's wrong because it's got a plus sign here. So they're adding these 2 matrices. you know, whatever. It's a minor detail, you know. Have you seen the cartoon faces, though, that this thing can make? You know, it's like, that's pretty amazing. So, unfortunately, though, besides these problems of maybe learning what NMF is, it's not quite what we need, okay? Because the structure of this matrix P is not just that it's positive, it's this doubly stochastic matrix. The rows sum to 1, the columns sum to 1, because we're trying to conserve the mass when we map points to points. And so that's a problem. NMF does not model this. And then secondly, we can't really quite use this NMF idea directly because the problem is we can't compute the P. It's too big. We can't actually compute it. So we certainly can't compute it and factor it. We need to do both things at the same time. So fortunately, a few years ago in the machine learning community, and building off actually some prior work in the mathematics community from a few decades ago, there's a derivation of what's called low-rank optimal transport, or low-rank factorization of this coupling. So what does that look like? So we have this big matrix P, describing how we move cells proportionally, and we factor it. Now there's this sort of intervening matrix. It's a diagonal matrix with these entries. And what's happening here? So Q and R turn out to describe a latent representation of the data, these latent points, so here 2 points. And G is this probability distribution over the points. Now, why do we have such a structure? Well, remember, this is all about mapping probability distributions. So the right way to factor this matrix is that we have to factor it into probability distributions. And so what we end up here with is a very beautiful structure. You get this soft clustering, if you want to think about it, of the data X into 2 clusters. So you've assigned each point with some proportion to each of these 2 latent clusters. And similarly with R. And then there's just a distribution, in this case, a distribution over 2 points, telling you the proportions of these 2 clusters. Okay? It's quite elegant. And you can, in fact, compute this factorization of minimum cost. Okay? So if I say, let me look at all the ways I could have found a coupling of rank R, we can find the minimum cost one, again with this entropy regularization, and we can do this in linear time and space. Okay? This is work by Marco Turi and his students, Setbon, and they describe how to do this. So this is great. So now we went from quadratic time and space to linear time and space. All right? So Fabian Thies and his group recently published a paper in Nature where they actually used this idea. So they combined this low-rank approximation, this low-rank OT, with some of the existing optimal transport formulations, so Waddington OT and low rank, Moscott time they called it, our paced algorithm in space, Moscott space, and then something they did for spatial temporal where they allowed for growth. And this allowed them to now produce alignments for data sets with these 80,000 different points. Okay? So you can't really tell, but they've aligned actually 3 different data sets right on top of each other here. All right, so this is great. This is low rank to scale, so we can scale to large point. But there's actually something a lot more interesting you can do with this low rank factorization. So let's go back to this Waddington landscape, all right? So we have cells sort of traversing down this landscape, but then there are also these, valleys in the landscape that we might term cell types. In fact, one of the first things people do with their gene expression data, their single-cell RNA-seq data, is they try to cluster it, and they try to find cell types, all right? So there's not just cells moving through space, there's somehow these cell types, and I guess I sort of messed up the animation on this. So we could think of what a lower resolution version of Waddington landscape is actually to describe the transitions between the cell types at each time point. And this is what we would call the differentiation map, okay? So this map has a vertex for each cell type. Somehow we'd want to identify those cell types, and then edges that describe which cell types differentiate into which other cell types. And if we're doing development, it's unidirectional, so these edges are directed. All right, so this low-rank OT gave us this clustering. So we've got cells here into these now clusters, which we can call cell types, and here. So here's at 2 different times. But it's not quite what we need, because what we want to describe for this differentiation map is transitions between different cell types at different times. And this representation sort of only has one notion of cell type that's shared across both time points. So it's not quite right. So if we think of the structure we want, it's something like this, where we've got cells that we want to cluster soft into cell types. We describe then these transitions between cell types, and then the cells at the next time point are somehow grouped and samples from those cell types. Okay? So what this led us to describe was an alternative factorization of this coupling matrix. This is called the LC factorization, previously described, and then we sort of worked out a new algorithm to compute this in a paper at NeurIPS a few months ago. So here's our coupling matrix describing the map between these distributions. We factor it into Q, which describes how to find this lower dimensional representation of the cells at the first time. Then this transition matrix T, which if it's a rectangular matrix, allows for different numbers of clusters or cell types at each of the two time points. And then those are then projected back out via R to the cell types at the later time point. Okay, and so we could compute this, and we could compute it with something called semi-relaxed OT, and you can look at the details for how to sort of do this. Let me just show you a visual of why this is important for a very simple data set. Okay, so here's a data set where, say at the first time we have these 10 clusters. These are just Gaussians arranged in this nice way. At the second time point, we have these 5 Gaussians arranged here, and then the optimal transport map would map each point to its closest Gaussian in space. All right. If you notice that the number of clusters at the two time points is different, 5 or 10. So if you run our algorithm, you can actually run it at rank 5, and you get a nice illustration of the latent points at the second time point being 5 and here, and everything maps nicely. If you run it at rank 10, you get 2 latent time points, two latent points for each of the green because they double up, but still it looks really good. If you run the other algorithm, it's forced to choose 5 points at rank 5, and so it's got these 5 latent points, the clusters here, but then it has to choose 5 at the first time point, and so it averages them. So it's like totally wrong, right? It's between the two clusters. If you ask for a rank 10, then it has to choose 10 time points at each, 10 clusters, sorry, at each time point, 10 latent points. So now it's got 10 here, and it also sort of messes up. Okay? So this number of cell types changing at the two time points becomes quite important to model. Now, at this stage, we could have published a paper where we took now this new factorization and ran it with Waddington OT or ran it with paste, and at 2 time points gotten probably very impressive alignments. But we wanted to do something more because we realized that a lot of the interesting data sets had multiple time points. So now at multiple time points, there becomes a small problem, because if you have the map from cells at the first time point to the second time point, you can describe it via this factorization. You've got now this clusters at time 2. Okay? But now from time 2 to 3, you could factor that one separately and independently find the clustering of cells at times 2 into cell types. And if you're lucky, maybe these two things were the same, but if you're unlucky, they may not be, but they should be because they're the same cell types. So what this says is that each of these factorizations at consecutive time points have to be consistent across the whole time series. Okay? So you have the same cell types at the middle time point, so the R here, the clustering matrix at the first to second time point, and the Q prime here from the second to the third time point should be the same. Okay. So this led us to develop something that, for reasons I won't get into, we call Hidden-Markov optimal transport. The idea here is you've got cells, spatial locations or not, you can do it with single cell or spatial data, at multiple time points. You find a latent representation at each time point into cell types, a grouping of cell types. This transition matrix between cell types, so here these two cell types differentiate into these 3, and later time point into T4. And now these transition matrices and the cell types have to be consistent across the whole time series. Turns out there's a really hard optimization problem, and we can't solve it exactly. So what we do, and this is sort of the inspiration for the name, is that we borrow some of the ideas that have been out there with Hidden-Markov models, with these latent representations, and there's this algorithm that goes forward in time and backward in time and finds the intermediate representation called the forward-backward algorithm, and we sort of adapt that in this setting, and so we were able to compute this with this forward-backward type algorithm. Okay, so unlike Pavel's talk, who said he was going to focus on all the biology, I've, in CGSI tradition, focused only on methods. I haven't shown you a single result. I just showed you a bunch of equations. So now let me show you a couple of results. First one's synthetic. We built this thing we call Waddington's volcano, I guess, just for fun. So there's one cell type at the beginning. It differentiates into 3 at the second time point, and then into 5 and 5. We simulate the whole Langevin dynamics of these cells moving down this Waddington landscape. And then we sample data, and then what should happen is that the data falls into the wells of this Waddington landscape, the cell types. So there's 3 at the first time point. Sorry, I said 5. It should be 6 and 6. So this is the differentiation map you should get. And we run our Hidden-Markov optimal transport. We do pretty well. There's a few edges here that are sort of stray. You can look at the underlying data projected here in 2D. Here's the data at the first time point. There's these 3 cell types. You can't really distinguish them very well, the 6 and the 6. If you instead try to use this Waddington approach, which doesn't have clusters, so you have to find the clusters, and so if you combine it with k-means, you get a much more messy construction. So if you look at the difference between the true and the observed differentiation map, our error is sort of consistently low as we increase the measurement noise, and it starts to increase pretty dramatically with the Waddington OT. Okay, so that's synthetic data. What about real data? So going back to zebrafish, here is data from spatial-temporal transcriptomics from 3 to 24 hours post-fertilization in zebrafish. So here's the cell types as annotated in the original data set. And so if you look at this, it seems very nice. They've got these beautiful cell types. But actually, if you start looking at their annotations, there's a few weird mysteries that crop up. In particular, there's this famous structure in development called the notochord, which is present at time point 10 and 18 in their data set, and completely missing at 12. All right? Now, maybe they cut the embryo in a different way at 12. Not true, the notochord should have been there. If you go instead, and why did this happen, is because they clustered each data set at each time point into cells types independently. If instead you link everything together, we can infer this differentiation map here I'm just showing at 10 and 18. We supervise it on their clusters at 10 and 18 just to make sure that we're not skewing things in a way. And we learn just the clusters at 12, and we find this cluster that is exactly linking the notochords. And the notochord's there. Okay, so they just missed it because of their independent clustering at each time point. And it's sitting here as this brown cluster right where it should be. Okay, so we recover the notochord. And there's a whole lot of other things we can do, but this idea of mapping clusters across time, we can of course learn everything de novo, but you can take knowledge of any cell types or clusters you have at any time point during the whole time series and push them forward or pull them back. All right, so that's HMOT, so these hidden or latent trajectories, this differentiation map that describes cell types and transitions. And so we optimize this low-rank factorization simultaneously across time points. The paper was presented at Recom. We're still kind of working on it in the journal version, but the code is there as well. All right, so in the last few minutes, I wanted to present something brand new we call hierarchical refinement, optimal transport to infinity and beyond. I'll come back to this in a second. And so the idea here was that, again, these data sets are growing very large numbers of cells. Here, 80,000, those mouse data sets, hundreds of thousands of cells. So we want to now compute alignments between these data sets in space and in time. And I showed you we could scale with low-rank optimal transport. That's one way to scale. But what would be even better is to compute at single cell resolution these alignments. In fact, that's what we really want. We'd love to know what exactly is the ancestor of this cell from this time point at the previous time point. Single cell resolution. That low rank approximation will not do that. It smoothed everything out over these low rank factors. So what we want is this Monge map, this like one-to-one mapping from cells to cells. And so what do we have? All right, so we can compute these Monge maps in cubic time using the old classic algorithms, matching algorithms. So now we have single cell resolution, but cubic does not scale. We've got all these low rank things on the other extreme that are linear time and space. So they scale, but the alignment quality be very poor. And so could we get something up here, this single cell resolution map in linear time and space? And so this is what the new algorithm that is being presented this week at ICML. This is why Peter and Julian are not here. They're presenting this at ICML. It got an oral presentation, amazingly, for the insiders. So some schlubs from Comp Bio, imagine to get the top 1% of ICML papers in the machine learning conference that has like 10,000 papers. The scale of that field is much different from ours. And so what this does is it computes these maps in linear time by computing them in this refined way. So we can go up to 1 million points, and there were some earlier kind of multi-scale OT approaches. But what Peter and Julian and Xinhao realized is that there's actually a nice theoretical connection here between these low-rank couplings that I described and these Monge maps. And I won't go into detail here, but I'll just sort of say that if you found the optimal rank 2 coupling, so rank 2 means I have to take all my data points and put them into 2 clusters, right? So I'm going to cluster at the cells at the first time into one of the 2 clusters, cells second time into the same 1 or 2 clusters. That clustering respects the best Monge map or one-to-one map. So it can actually compute at the first time step the optimal mapping between a partition of my data into 2. Okay? And then that idea iterates. So we can actually use this low rank, find the clusterings from them, and then iteratively learn this Monge map at finer and finer scales. And that's what the algorithm does. You get the Monge map between partition into 2, and then you repeatedly, recursively refine these partitions until you get down to block size 1. And then you get linear time, space, linear in space, and linear in time. Assuming your cost matrix, you can compute in linear time and space. All right, so just sort of one example. Here's an 84,000 by 84,000 Monge map between these 2 mirrorfish data sets. This is a spatial transcriptomics technology with single cell resolution. And we can get these Monge maps, and then we can carry forward and see, at one time, how well could we, predict the expression at the next time slice, and we can do pretty well. All right, so still very much in progress in terms of the applications, but I'll just show you sort of the scalability. This is on a synthetic data set, and so they are scaling, up to 1 million. And I just put this here because, we had a debate. They wanted to title this paper "To Infinity and Beyond" to be kind of cutesy. And I said, guys, 1 million is not infinity. You haven't reached infinity. And I just don't have the pull in my group that I used to. So you see who won in terms of the title. So that's hierarchical refinement. So now we can compute these single cell resolution maps for infinity cells, I guess, if you believe them, 1 million if you believe me. And this allows us to now push this forward into the bigger data sets that are coming. And so I think there's a real opportunity here for this community to really crack one of the biggest questions in all of biology, which is, what's this whole process? And you can imagine that the data that's coming is from many time points, spatial, temporal, multi-omics data. It doesn't have to be just gene expression. There's spatial attack-seq. There's all this other stuff. There's, from the lineage side, spatial lineage tracing coming. And of course, you can do perturbations on all these things. So I think we can all become developmental biologists if we want, at least fool people into thinking we are. So with that, I'll end and just acknowledge the group who provided lots of input into this, and Julian and Peter and Xinhao who did the recent work. Earlier, Ron Zira is the one that really got us started on optimal transport with paste, and it's been an interesting few years, and we're still pushing on it. So thank you for your attention. I think I finished on time, so if there's happy to take questions. Yeah, so I think the question's about how to handle disproportionate cell types. I think for like the low rank part, right? So you've sort of hit on one of the biggest challenges, which is how to kind of what are the hyperparameters we use for things? What's the ranks that we use? And so we can, of course, we have to choose the number of cell types to do the rank thing. So we can choose those. We've thought about different ways to supervise it. So when we do this de novo, at least with current data sets and the way we're processing gene expression, it's hard to know the number of cell types to choose. It's hard to know what parameterize. We've thought of different ways of supervising it. One of them is, of course, to use some clustering from other time points so we can carry forward, as I described. The other is if we have information about just number of cell types and maybe some estimate of proportion. This seems to help a lot. And the reason why is, the sort of classic problem that if there's, 10 cell types that have wildly different proportions, including some rare ones with like, 1% of cells, and we just ask for 10 cell types, we'll end up probably dividing a big cell type and that we won't find the 1%. So this kind of proportion supervision is something that we've been playing around with and has been somewhat successful.
Yeah. Similar question. I'm thinking about like how densely you sample across time to not miss intermediate states.
Yeah.
If you have some like prior on the cell differentiation map, if you can use that to kind of fit with fewer time point samples.
Yeah, I mean, that's a great question. So how densely do we need to sample in time? I'm not a developmental biologist, so I can't, perhaps give you the best answer to this. There's, of course, if lots of stuff happens between our two time points, this idea of finding these minimum cost maps between them they're only going to be interpolate so much. So they're not going to somehow magically produce intermediate cell types that were not measured. That's a severe limitation. I would say if you want the method to work robustly, you need dense enough time points so that there haven't been too many unmeasured cell types. We think that movement is movement in the slice is something that we can control pretty well, and the gene expression distance, a little bit okay. That's sort of where this. If there's other cell types, the gene expression distances start becoming kind of wild. So, yeah, I mean, I don't have an exact answer, and I'm sure it would vary based on any particular organism and how quickly the cell types are transitioning. But that's, I think, something that we're actively working on. I didn't sort of present this part, but, where you can interpolate between these maps, and we're doing that. And so you can interpolate, then you can extrapolate. So we can make predictions, and we could start to perhaps get handles on the experimental design problem when we do that. And I think that's a great direction.
Yeah, thanks for the great talk. So I'm trying to think if the low rank is a good assumption for coupling between these slices. So in the simplest case, if the slices are sort of identical at the two time points,
Yeah.
your coupling would just be an identity matrix, which is not low rank.
Yes. Yes.
So how does it work if I have very identical slices? What would happen running this kind of a method?
Yeah. So this is a great question. Is the low rank the right approximation? Because obviously the identity matrix is not low rank. That's exactly why we started to switch to these Monge map formulation, because we don't think it's actually the right thing to do. In that the data is not low rank at the single cell spatial level. So at the spatial level, every cell had a unique spatial location. Now, it might be low rank at the gene expression level. So there's sort of this disconnect between what you want for the two measurements, but the actual coupling matrix from cell to cell should not be low rank. And so yeah, you could use low rank for scalability, as was done in the method I presented, but we don't think that's actually the solution for spatial data, where unique spatial locations. Maybe for single cell data where all you have are gene expression, you have cell types, there it might be reasonable. Maybe I'll take one last one. There's no moderator, so we'll do the last one and maybe go to break.
So when you show that the optimal transport is in the spatial domain and also expression.
Yeah.
Now, somewhere in your
Right.
And I guess my question is it first robust to the choice of that parameter? And second, is there anything interesting we are learning in terms of how much the space versus expression tells you about Wilson system?
Yeah, great question. So there was this balance parameter alpha between space and expression, and so it was a convex combination of the two. And how do we set that parameter? Well, it's the usual hyperparameter selection thing. But how robust is it? It's robust to medium-ish perturbations in alpha when we've looked at data sets. But of course, you can skew things toward expression or toward space by changing that parameter. We have parameters that we like for data sets we've looked at that we tend to sort of stick with. But we've seen that depending on the gene expression measurement, and sometimes they're whole transcriptomes, sometimes they're like these, a few hundred targeted genes. The balance of how important the expression signal is, how much information the expression signal carries versus the spatial signal can change. So I don't have an answer, I don't have a great answer. I also think that probably the answer is not a convex combination of those two separate similarity measures. And I think there's a lot of work to be done to actually kind of do this in a more rigorous way with some sort of genuine spatial temporal model. So our first attempt, but a lot to be done there. All right, let's go to break.